Hello, my name is Jeff Martin, part of Facilitate TV, and I am pleased to be joined by Rafa Agnar, the co-founder, president, CEO, titles of NKILT Therapeutics,
Yes.
based down in Houston. Rafa, what are you guys doing over there? What's going on?
Well, thanks first for having me. It's a real great pleasure to be part of this discussion. And so we are developing a novel cell therapy approach focusing on allogenic technology to be able to bring an off-the-shelf, ready-to-use cell therapy. And more importantly, the goal is to build a technology that can be scalable, so they could become more accessible and then down the road profitable for the developers.
Right.
And not to minimize the importance of finding treatments for patients, I am always a believer that if you have a treatment that can be accessible to more and more patients, you will be generating profit at the end. So it's really kind of the definition of what we're trying to do. And we have a technology that we started because we're going allogenic. We started it with NK cells, but actually our technology can be applied to any type of immune cells. We could do T cells, we could do gamma delta T, NKT, but we decided to go first with NK cells and build a platform around the NK cell.
So I'm a huge fan of NK cells, and the name of the technology is in the name of the company.
Exactly.
ILT, so it's a chimeric ILT receptor based system. What is an ILT receptor?
So the focus of NKILT and the idea behind with my co-founder was to focus on one target called HLA-G. And HLA-G is actually known as the pregnancy molecule, is what actually the fetus and the placenta are using to shield the fetus from the immune system of the mother, because it's kind of a foreign entity.
Sure, yeah.
So it should be under attack of the immune system. And the hypothesis is that actually HLA-G for 9 months is protecting the fetus from these attacks by shutting down the entire immune system. So if T cells come any close to the fetus, they're not going to attack it, making it like more or less that's the story. The thing is that cancer cells, over half of cancer cells across all type of cancers are expressing HLA-G, and they kind of use it as a blanket to shield from the immune system. And what they do is that they shut down the immune system, so you can recruit T cells, NK cells, whatever, to go to the tumor. But if HLA-G is shutting them down, nothing is going to happen.
Yep.
So part of our hypothesis is that we believe that we can really enhance the immune system by blocking HLA-G. Now the trick, and you mentioned the chimeric ILT receptor, people say, well, you're not using a CAR.
Yeah.
Well, the reason is because HLA-G is expressed in 7 different isoforms.
Okay.
So if you use a CAR with an antibody antigen binding, it's going to be great when the cell are going to express HLA-G1. But when they're going to start to express HLA-G2, HLA-G5, then your antibody is going to be challenged, because by definition, an antibody with an antigen is very specific.
Sure.
The key here is that instead of using the antigen to bind, we're using the receptor, which are the ILT receptor, which are the inhibitory receptor that are triggered by HLA-G and that triggered that immunosuppression from HLA-G.
So would it be fair to call it a switch receptor? It's kind of like a
I would not
switch.
use the term of switch, but it's kind of we're using a receptor binding instead of using an antigen binding.
Yep.
The trick is that the receptor is activated by HLA-G regardless of the type of HLA-G.
Sure.
So we actually had some interesting discovery. For example, as an anecdote, one day we were screening a HLA-G negative cell line.
Okay.
And we were seeing responses from our cells.
Uh-oh.
And then we discovered that it was an HLA-G1 negative, but it was actually heavily HLA-G2 positive. And our cells were recognizing it.
Yep.
And a standard CAR targeting HLA-G was not.
Right.
So this is part of the reason why we decided to go with the receptor binding, because we think it will be a better way of targeting HLA-G.
So then I have to ask, if it's immune cell type agnostic, then why NK cells first?
So that's a good question.
I have my theories, but I'll let you answer for yourself.
So the first reason, which is kind of the obvious one, we're going for an allo approach. NK cells are probably the easiest candidates to develop an allogenic technology. But when we started working with it, a lot of people say, oh, NK cells don't work, they are lazy killers, whatever. yeah, whatever. T cells are way better. First, I don't like to make comparison between different type of cells. They all come with their own benefits.
Yeah, our body has them for a reason.
I mean, we
right?
have so many of them. It's like,
Yeah.
So yes, CAR T is great, but my dream is that one day maybe we'll have a product where we have T, NK, iNKT, whatever it is, because
Sure.
they all have a role to play. Now, this is probably impossible to do technically speaking and probably regulatory
Currently.
wise.
Well, sure, yeah.
But NK cells also come with a lot of benefits. If you find a way to activate them properly and to grow them properly, which we can do, some people think it's challenging. We don't think so, actually. But if you can engineer them properly and really activate them strongly, they can become very strong killer. We really call ourselves serial killers of cancer because they are.
Yeah.
We had very great data that we presented at ASH in 23 as an oral presentation, for example, which was our in vitro proof of concept in AML. And we had incredible results because of our secret sauce in terms of activation.
Sure.
But the other thing also is that NK cells, people will say they die out after 2 weeks. True. Unless you have some nice activation approach that allow your cells to stay active and killing for up to 6-8 weeks, which is what we have done with our technology. It's part of our IP that we have submitted. It's actually a second IP, the first one being the construct.
Sure. See, and I think that, we just saw a talk about the challenges of allogeneic T cells. One of the big things is the elimination of CD3.
Yeah.
Right, and it's just like a whole step that's not required for NK cells because they're not alloreactive like that.
They're not. And then people are going to say, yeah, but at some point you're still going to have rejection. Yes, I agree, but it doesn't mean that
Same with
that's not gonna
T
work.
cells, though.
With
Yeah.
anything you're going to bring.
Yeah.
The main thing is that in the window you have before the rejection occur, how strong and how good are you going to be in attacking and killing cancer cells?
Yeah.
Hence the serial killing. You need to have very active, potent cells that are going to go. The other benefit of NK cells that I want to emphasize are two other key things. Number one, from a safety profile standpoint, you have significantly less, and even sometimes almost
None. Yeah.
cytokine release reaction,
Yeah.
which is a great benefit.
And it's a characteristic of the fact that they're like the weaker killers. It's like,
Supposedly.
Yeah, well, they're also less, crazy.
Inflammatory.
Right? Yeah.
And so the interesting thing is that it's all about balance. You need to have a little bit of reaction, but not too much. And again, we have grown dramatically in making T cells significantly less inflammatory with cytokine release. So we know how to do that through some nice engineering or gene editing and whatever. Okay, but it's just kind of it's right there. It's a natural approach that we want to use. The last thing that I think people underestimate is the innate killing properties.
Exactly.
Because without having any targeting, they already have that kind of killer instinct.
Unlike T cells, which are very, very specific.
And I will also say, unlike antibody technology, because an antibody is an antibody. It's gonna bind and trigger the
it's
killing.
very vertical. Yeah, in that sense.
And it's great.
Yeah.
And you can really have incredible examples in our business of extremely successful drugs, no doubt about this. But that additional innate killing, for me, brings a kind of another push that can play a role down the road when we use a treatment like this.
Yeah. So I have to ask, the big hype these days is in vivo delivery.
Yeah.
I know that I think it was Interius who was recently acquired by, I think it was KITE. Their approach was a CD7 approach, and you can get in vivo CAR T, but also in vivo CAR NK cells.
Yeah.
Are you guys threatened by the in vivo approaches? I have my opinions on in vivo, especially in oncology. It's not going to work in oncology, I don't think. So how do you guys see the threat from in vivo?
So I don't see it as a threat, actually. Number one, we need every type of approach to beat cancer. Okay, if people believe that they're going to have the secret sauce and their thing is going to eradicate cancer, good luck with that. I heard that many times.
Sure.
And how many millions of people are dying every year? So we're not really good at it, I guess. Now, we made significant progress because of the variety of options that we bring to patients. Because by nature, the cancer cells are gonna fight the hell out of you to survive. That's what they do. They survive.
Yep.
They find any trick to survive. It's the same way the normal cells will do everything to survive.
Well, evolutionary, progression is going to do it.
So that's the first issue that I never see a technology as a threat. I see it as a complement. We all need to come to the table because patients need options for them, and they obviously don't have enough. We need more. The second piece is that I actually really see it as an opportunity because, to the contrary of what you just said, I believe it can work in oncology.
Okay.
The thing I don't believe is that it's not gonna work tomorrow.
Right.
It's gonna take time.
The current iterations of
we're gonna have some successes. I am convinced. Like actually, you mentioned Interiors. I really like what they're doing. We know very well this company. We look at them. Brilliant people, brilliant ideas. Capstan also very good. Like for the one that was acquired by AbbVie, whatever. A lot of very smart technologies, but when we created CAR T, it didn't happen overnight.
Yeah.
In vivo is not going to happen overnight either.
Yeah.
It's going to take some time. Like I said, we're going to have few wins, but there are a lot of key questions that we need to answer and that we haven't yet answered. And that's our job.
Right.
Now, how do I position myself and NKILT with this? Well, there are two ways of looking at it. One is that we are on our lane for allogenic technology and we believe in it and we're going to pursue that. But I'm not stupid. There is clearly a trend and a higher interest of in vivo because it is the future in some way. It is part of the future of cell therapy.
Yeah.
And we are working very hard currently with my team on identifying how to apply our KIR technology to in vivo.
Yeah.
And I can at least say that we have found a path.
Okay.
But I don't know if it's NKILT.
Sure.
Maybe it's something else.
Something new.
We'll see.
Yeah.
But I am convinced the KIR could be part of the equation if we wanted to work on it.
Yeah, that's interesting. See, I hadn't thought of that. I mean, like, if you're saying that the HLA-G is, is it mainly a solid tumor thing, or is it also in heme, or
everywhere?
Is it everywhere, right?
It's expressed across all type of cancers.
Right,
so.
And it's a massive, like, stealthing, quote unquote, mechanism of the
and it's really about the thing about HLA-G is, I mean, we used to say, the target is really key, whatever. It's actually not the target that is key, it's what the target triggers, is what it is involved in.
Yeah.
And really what is involved in is literally shutting down the immune system, protecting the tumor, and in some way, it's also really involved in those tumor escape mechanisms. And so if we use it as a gate to kill, but there's a lot of other things that hopefully we'll be able to prove that will be a natural cascade of event that can go from bringing the tumor microenvironment back to a more inflammatory status, so then you can have actions of other drugs like PD-1, for example. But there are a lot of hypotheses that us and many other people are looking after. So I think there is a lot of interest in building more science around it. And for me, in vivo allogenic, it's kind of the vehicle that you're going to use to reach the tumor.
Yeah, right.
Are there some that are better than others? Probably. But we will only know when we put them at test.
Sure.
At the end of the day, it's in the clinic that everything is going to show whether or not it works. This is part of the challenge we have faced in cell therapy recently.
Yeah.
Is that a lot of very smart technology looked really good preclinically on animal models and whatever, and then suddenly you hit the patient and what you have seen in animals is not necessarily replicated in human beings, or not to the same degree. And well, that's medicine,
Yeah, especially in oncology. I mean, I'm a cancer biologist by training, and the models we have available for preclinical studying in cancer are just not really working at the moment, I think.
Well, it's
the translation is just never predictable.
We always use that old adagio, is like we have cured every cancer in mice.
Right. Yeah.
Even recently, there was a very nice publication from that group, I think a Spanish group like that. They just said that group found a way to cure pancreatic cancer in mice.
Yeah.
Yeah.
Congratulations. How about humans now?
And we need that. But how long is it going to take to replicate that in human?
Right.
We know it's going to take a long time.
Or can it replicate itself in humans?
I want to hope that it can, but then again, the use of cure for me is something that we
Right.
need to be more cautious about.
Right. So then last question, what do you think in terms of the industry at large, what are like, 3 things, let's say, that you expect to see in 2026 around like cell therapies?
So I will have an optimistic view and a pessimistic view.
Okay, let's hear
To
it.
try to look at both.
Sure.
I'll start with the pessimistic. I think we're not going to see, unfortunately, a real push up, a real restart of acceleration of new technologies and whatever. I think yes, in vivo is going to continue to be a big hype, but maybe we're going to see some early signs of things that will be interesting. But I believe it's going to ask probably more questions than it's going to give answers at this stage.
Which is good. I mean, it's good to ask questions.
We
get
need
the
to open
answers.
doors.
Yeah.
But the correlate to this, which is a potential issue, is that I don't see funding coming back strongly in early stage. I think we're still going to be in a year of mega deals. We're still going to be a year of consolidation and capital investment instead of venture investment.
Sure.
And I still see that maybe the IPO window in the US is going to reopen. It looks like it's reopening in Asia. So maybe it's going to lead with some kind of dynamics on that front. But I think it's still going to be, financially speaking, a very challenging, but better than last year. The positive, that's going to be the half full piece, is that I'm actually extremely excited on the amount of novel approach that are coming through. There's a lot of great science coming. How do we convert that in great treatments?
Right.
That's the key thing. How do we work better together to not repeat the mistakes that we have done? How do we share properly some of the information? I know we cannot share everything because, IP and all this. Fair point. But instead of fighting against each other, if we share some of the learnings that we did on our successes and on our failures, we have more chance of building the treatments that the patient needs tomorrow. And I know that many of my peers, they have the intrinsic will to do it. Now it's very difficult to pull off because you need to align a lot of people behind you on that. But you have to start somewhere. And sometimes just a little collaboration on something can make a big difference.
Yeah.
And I'm not saying that we should all merge in a big entity that's going to develop cancer drugs.
Right.
No, because we still need that diversity. But we really need to use platforms like the Advanced Therapies Week meeting or other key meetings in the field of cell therapy to really share our best practices, the good, the bad, the ugly, and realize, hey, you know what? They did this, it didn't work. I think we know why. Let's make it work.
Right.
And if you succeed where I stumbled, if it leads to a patient receiving a treatment that can be life-saving, then we are doing our jobs.
Right. Yeah.
Of course, I want it to be me and my company.
Well, right. Yeah. It's all about the money, but
but it's at the end we have to remember.
Exactly.
I am still a little bit of idealistic. We do this for the patient. If I was doing this for the money, I would have stayed in big pharma.
Sure.
I would not have created my company.
Sure.
So it's not about the money. Some people will say it is a little bit. Yes, maybe. Hopefully, it will be about the money. But like I said at the beginning of our conversation, the money will flow if we do what we're supposed to, which is to bring new treatments to patients.
Exactly.
That's the only thing. Now, the last comment I want to make, because it's the elephant in the room. We live in a very strange and I would say hectic political environment globally.
Sure.
I'm not going to play politics here. It's a global situation. That doesn't help. In the US particularly, there's some strong headwinds against science, which are very difficult. I'm tired of having people ignoring it. We can't ignore it. This is hurting us, and it's hurting the patients down the road. We have to work on that. The center of gravity of cell therapy has moved east. It's a given. The leadership in cell therapy is moving east. We need to accept it, and we need to surf that wave, because it's only together that we're going to make it.
Sure.
If we battle for stupid politics, at the end,
patients are going to
lose it.
Yeah.
So it's our duty as leaders in the field to find a way to still make this work, regardless of the political environment, which is hard. And if you cannot control it, stop fighting it.
Sure.
Just
That's one mechanism.
Just find a way
Right.
to go through it.
Right.
It's our responsibility, I think, as leaders in the field to find a way.
Yeah.
And I believe that we will find a way together and not on our own. That's my belief.
Sure. All right. Well, with that, Rafa, it was awesome talking to you. Thank you.